In this movie, we consider the mechanisms of contraction of striated muscle, in particular, the arrangement and interactions of actin and myosin. There are four basic features and processes involved in the contraction of muscle. The first is the structural features of actin and myosin itself. The second is the role of ATP and how ATP powers the interactions of actin and myosin. The third is how the action potentials that sweep through the transverse tubules cause the release of calcium from the sarcoplasmic reticulum. And fourth, the role of calcium and how calcium allows myosin to interact with actin. We will only discuss the first two features in this movie. Features 3 and 4, the roles of the action potential and calcium, will be discussed in the next movie. So we're going to start off by considering the muscle itself, which is shown here in the top of the figure. The muscle is actually composed of many cells called muscle fibers. Notice that each of the fibers has an alternating series of light and dark regions, and thus each has a striated appearance. Each muscle fiber, in turn, is packed full of structures called myofibrils. Each myofibril, in turn, also has a striated appearance. A magnified view of a myofibril shows that striations in more detail. Notice the regular arrangement of the black lines labeled as Z lines. Between the Z lines are two dark regions, shown as horizontally arranged orange lines, separated by a small white region. The region between two Z lines is called a sarcomere and is the unit of muscular contraction for reasons that will be explained next. Here we see the arrangement of the two major muscle proteins, actin and myosin, as they're arranged in the sarcomere. The thin actin filaments are anchored to the Z line in a perpendicular arrangement. It is as if these actin filaments are actually welded to a solid vertical structure, the Z line, and are oriented perpendicular to the Z lines. Sandwiched between the actin filaments are the thicker myosin filaments. This arrangement of actin and myosin explains why the sarcomere has a striated appearance. The region just next to the Z line has a light appearance because there is only the thin actin filaments in this region. That region is also called the I band. Next to the I band is a darker region shown as alternating orange lines. The reason that it is darker is because here the myosin filaments are sandwiched between the actin filaments. This region is also called the A band. Notice the light region next to the darker A bands. That region is lighter because it contains only myosin filaments. This central light region is called the H band. Thus, each sarcomere has striations because of the arrangement of the actin and myosin filaments. In the next movie, we consider how the interaction of actin and myosin are regulated.